Welcome. Imagine a world where you can combat disease in advance. Wouldn't that be brilliant? You could basically see into the future. Well, in fact, this is possible thanks to the CDC's new Genome Project. Let's explore the origins of Listeria and get to the root of prevention. Hello, I'm Adrian, your foodborne illness investigator. Listeria has caused 1,600 cases annually according to the CDC. Could whole genome sequencing be the solution for reduction? Let's find out. Today's session will consist of the following breakdown, starting with an introduction and background of the origins of Listeria, the methodology utilized for whole genome sequencing, results using those methods, and the conclusion of those results of how we can utilize this data to continuously reduce outbreaks of Listeria. About 90 years ago, Listeria was formally named as bacterium monocytogenes by E.G.D. Murray. After Murray observed gram-positive blood in rabbits, he attributed this name. By 1940, J. Harvey Peary officially labeled this bacterium as Listeria monocytogenes due to its catalase-positive properties. In short, a bacteria is considered catalase-positive due to possessing the enzyme catalase when bacteria is added to hydrogen peroxide and gives off a gas. Historically, Listeria's first outbreak took place in Germany in 1949, specifically targeting newborns. Shortly after, 6,000 samples were collected from animals and environmental sources. Investigators then concluded Listeria was in fact in these samples and had the possibility of being present in both humans and animals. Of course, later we entitled this result correlation to zoonotic transmission. Continuing. Considering Listeria is present in cattle and poultry, the food industry items that are common with Listeria are deli meats, soft cheeses, unpasteurized foods, chicken, and decaying produce. Listeria has 8 species that make up its genus and 16 serotypes that contribute to 90% of human infections. It also has 4 lineages. Lineages 1 and 2 commonly in humans, lineages 3 and 4 found in animals. Conditions that Listeria thrives in are 40 degrees Fahrenheit and below. They can grow with or without the presence of oxygen and between a pH of 4.0 and 9.6. The diagnosis of the Listeria syndrome is listeriosis. In terms of the epidemiological breakdown, 1,600 people are diagnosed with listeriosis annually. 1,460 are hospitalized and 260 succumb to listeriosis. Those considered at high risk for this infection are the elderly, whom often experience chronic sequelae such as septicemia, meningitis, or death. Immunocompromised is another group at high risk, with similar symptoms. And AIDS patients specifically experience 300 times more likely to be infected with listeriosis. Infants are the next at-risk group, and pregnant women. Listeria presents a significant economic burden, as you can see from the figure above, ranking second to Vibrio vulnificus in costs and death rates. Listeria is actually ranked first in terms of hospitalizations. These epidemiological and economic burden outcomes clearly indicate that Listeria has public health relevance and prevention will be essential going forward. Clinical signs and symptoms. Symptoms onset is 1 to 4 weeks and it varies based on anatomy that has been infected and whether the patient is pregnant. Pregnant women encounter symptoms similar to influenza, including body aches, fatigue, and can lead to spontaneous abortion, premature births, or even congenital transmission of infection. Others experience headaches, neck stiffness, and poor coordination. Let's discuss the big concept of the Listeria whole genome sequencing project. The process of whole genome sequencing involves 3 steps. Step 1 involving medical staff collecting samples from patients, the FDA and the USDA collecting samples from food, local and state officials collecting samples from farm and food industries, followed by food scientists actually identifying the whole genome sequences, and finally comparing the genome sequences to see if the banding patterns are clustering or similar. The outcome of whole genome sequencing provides a vivid analysis of which strain led to which infection. It detects strain similarity among individuals. Typically, the same strain may indicate the same source of contamination. Public health investigators with whole genome sequencing can detect more Listeria outbreaks in one concentrated area, connect Listeria to a causative agent, find new sources of Listeria, and halt Listeria at its early developmental stages. The image above reveals the overarching goal of this project: to solve more outbreaks, causing a significant reduction in cases. So, now that we know how to demonstrate whole genome sequencing, what can we do to organize our data? In terms of specific methods, the whole genome sequencing started in 2013 with the collaboration of the CDC, the FDA, the USDA, the National Center for Biotechnology Information, and state/local health departments. They all got together to compile this information. The sources of data were extracted from PulseNet, the Listeria Initiative, and the Genome Tracker. Let's analyze what each method consists of. PulseNet is an active surveillance system and national laboratory network that gathers data about Listeria and other pathogens. Utilizing the pulse field gel electrophoresis process, the Listeria's genetic fingerprint can be determined. Above, you will see the various steps that start with bacteria cells being taken from an agar plate and mixed with agarose, followed by biochemicals breaking them down and lysing the DNA into fragments. The fragments are then stained with gel and the patterns are observed in UV light. Going forward, the PFG produces a genetic fingerprint with the patterns, and depending on the patterns, we observe whether they have similarities to see if they're from the same source. The Listeria Initiative is a nationwide epidemiology system that gathers information about infections in humans. Information for this system is obtained through PulseNet. PulseNet compiles clusters of Listeria outbreaks determined from questionnaires given by physicians. This initiative works with the CDC and local health departments, and their objective is to reduce the time it takes from the outbreak to the intervention. The image above shows a timeline from when the individual gets sick, eventually experiences symptoms, goes to the doctor, has their lab tests performed, whole genome sequencing take place afterwards, and from the comparison of bands of various patients, see if they're determined to be a case. Genome Tracker. Similarly, the Genome Tracker is a nationwide laboratory network that makes use of PulseNet data to connect Listeria to DNA fingerprints from patients. Genome Tracker consists of the CDC, the FDA, along with college institutions and labs. In their efforts, the CDC is mainly sequencing patient samples, and the FDA and state divisions are sequencing food samples and also samples from the environment. To the right, you'll see a map that basically displays the dispersion of various genome tracker labs all throughout the United States. So let's get into a real-world example. Various research has proceeded after establishment of the whole genome sequencing process for the prevention and reduction of Listeria. For the sake of time, I will focus on one investigation carried out in Washington State. Utilizing PFGE and various forms of whole genome sequencing, isolates were extracted that all linked to ice cream. Remember that banding pattern we discussed earlier using PFGE? Well, this is a vivid representation. Samples were collected from Hospital X, Company A, Company B, and various local clinical facilities. Results from PulseNet indicated clustering on the gene ASCI. These banding patterns linked to Company A and Hospital X. Company A actually provided the ice cream to Hospital X, confirming that Hospital X milkshake machine was the source of the outbreak. Whole genome sequencing in this study remained consistent with the CDC's approach. It managed to trace pathogenic strains of Listeria in 2 different facilities within a year. Just imagine how quick a turnaround of investigation could take place for future cases. Immediately after the outbreak, preventative measures were taken, including routine cleaning of the ice cream machine. However, I found these methods unrealistic because Listeria is really difficult to simply clean up and remove. Instead, removing the source of contamination would be the best method. Limitations in the future. Limitations that the investigators run into is the high expense. It's usually over $1 million to conduct whole genome sequencing, and all variants sometimes aren't detected, some genes are missed, so there's a lot of work to be done. For the future, there are some positive aspects of the whole genome sequencing. When it's actually covering a specific region, it covers typically 98% of the coding region, and variants can also be detected in a non-coding region. What we want for going forward for the whole genome sequencing is consistency with detection so we can continue to solve outbreaks. Thank you so much for your attention, and if you are interested in your leisure to explore this topic further, the CDC has an investigation outbreak app provided for Android and Apple phones as well. Please check it out. Listed below are my references.